My name's Dr. Terry Lawton. This is old version, but anyway, I'm Director of Research for Reading and Cognitive Remediation. And does anyone here work with children or adults who can't pay attention or read? Our research has found that repeated retinal stimulation that remediates visual timing improves attention, reading, and memory significantly in dyslexics. The major problem we face in our school is that 80% of low-income fourth graders and 66% of all fourth graders in the United States are not proficient in reading, according to the National Center for Educational Statistics. Our research shows that these reading problems are likely to be a result of visual timing deficits. These visual timing deficits prevent attention and reading networks from developing properly. They prevent Common Core from being learned. When reading, students who allocate all of their resources to identify the letters in the word instead of interpreting a sentence, understanding its meaning, and integrating information into existing knowledge need visual training exercises to remediate these visual timing deficits. Training on left-right movement discrimination is a tool for the mind that remediates visual timing deficits. Therefore, we developed Path to Reading training, and it was designed to optimally activate magnocellular neurons that are signaling motion relative to parvocellular neurons, which are signaling pattern information, to remediate these visual timing problems. How does this repeated retinal stimulation work? When using patented left-right movement training exercises, we have found changes within weeks that provide lasting changes in attention, reading, and memory. Path to Reading training is needed so that the letters in a word are identified automatically, enabling Common Core to be learned more easily. It is a computer program that is rapid, taking only 10 to 15 minutes twice a week, and when done before guided reading, remediates attention, reading, and memory. Now, what is the stimulus that's needed to detect dyslexia? When the direction of motion is discriminated relative to a stationary background, then all types of dyslexics have deficits in movement discrimination. This pattern combination, movement relative to a stationary background, activates magnocells relative to parvocell activity. The parvocell activity being used to set up the background frame of reference for discriminating movement. So here's an example. I don't know if we can, it doesn't seem to be, is there like a,
you can move the mouse pointer over. Move it to the right.
It doesn't seem
all the way to the right, and it'll show.
All the way to the right?
Yeah.
Oh, good. Perfect. Okay. Now, these are sample patterns that we use. This is a single frequency background, and this is a pattern that dyslexics have a great deal of difficulty with because the test blends into the background. And these patterns here are multi-frequency. They have, and you can see they provide a wider frame of reference. So we show, what is the paradigm that we use? We show a test frequency against different backgrounds. We measure the ability of the subject to detect leftward versus rightward movement as the contrast of the test frequency is decreased until movement is discriminated correctly 79% of the time. So we're measuring contrast thresholds for left-right movement. We repeat this process for many different test and background frequencies. There are 4 different test frequencies, a quarter, half, 1 and 2 cycles per degree, and these are optimum for magnocellular neurons, and they also have to be low contrast because the magnocellular neurons saturate at 10% contrast. Now, each one of these test frequencies are shown against 5 different backgrounds. being 4 times wider, twice as wide, equal to the test frequency, twice as narrow, and 4 times more narrow, each being an octave apart, since the spatial frequency channels are spaced 1 octave apart. There are 16 levels of complexity. The complexity level was increased by increasing the number of sine wave components in the background, providing the frame of reference from 1 to 3, the background contrast from 5% to 10% to 20% contrast, recruiting more parvo cells at higher contrast levels, and 3, the pattern's speed of movement increases after every 4 complexity levels, increasing from 6.7 hertz up to 13.3 hertz, which is the peak temporal frequency sensitivity of the human visual system. We found that movement discrimination contrast sensitivity increased significantly as the complexity level increased. So you can see here the complexity level going from 1 to 16 for each of the 4 different test frequencies. This graph shows the pooled data from 2nd grade students doing the PATH training in the San Diego schools. Multi-frequency backgrounds confer an advantage when discriminating the direction of motion that we found from this. And you can see it here. I seem to have trouble with this thing. So you can see it here by at level 2, at level 6, and level 10, and level 14. That's when it goes from a single frequency background to a multi-frequency background. And we found across subjects an improvement in the contrast sensitivity. Now notice that the lowest line, the blue line, the quarter cycle per degree, is the least sensitive, and the reason for that is a quarter cycle per degree requires cooling across spatial frequency channels. Blakemore and Campbell in 1969 showed the lowest spatial frequency channel is to 1 cycle per degree, and we found the green line was the 1 cycle per degree, and we found the largest improvements in contrast sensitivity at each complexity level, averaging about 11.2% increase. Finding an increased contrast sensitivity function at increasing levels of complexity at all test frequency suggests that direction discrimination training improves the functioning of magnocellular relative to the functioning of parvocellular neurons. Not only was contrast sensitivity for direction discrimination increased significantly following this left-right movement discrimination training, but also the time to discriminate the direction of movement was reduced significantly for students who did the PATH intervention. We found that, okay, we'll get the next slide, sorry about that. Mouse has disappeared. Mouse to the left. All right, there to the left, okay. Sorry about that. Okay, we found that left-right movement discrimination training significantly improved attention, reading, speed, and visual and auditory working memory. We compared 2 different reading interventions. PATH are the blue bars and Fast for Word are the striped bars. Compared to controls who did learning upgrade in the classroom, Fast for Word changes the neural timing deficits in the auditory system. A lot of people say that auditory deficits are the basis of dyslexia, and our data proves otherwise. And PATH changes the neural timing deficits in the visual system. So Fast for Word takes over 2 to 4 times longer to administer, yet PATH increased standardized scores significantly, whereas Fast for Word did not. These graphs show improvements over controls who were trained on learning upgrade, these results being significant out to 2 and 3 decimal places. For example, attention and reading improved significantly at p less than 0.004. We also found that the more PATH is trained, more reading rates improved. As we increased the number of replications from 0 to 26, the reading speeds improved from no improvement up to 11 times faster. We plotted the final reading speed divided by the initial reading speed. PATH training has been proven to be faster only 15 minutes, 1 to 3 times a week, then other interventions, and can be administered to an entire class of students in the computer lab or classroom, unlike other reading interventions, which is what we're doing right now. Now, this figure shows that the improvements in reading skills after left-right movement discrimination training in second grade are sustained over time when looking at the same students in third grade. No one's reading skills degraded over the summer between second and third grade, and when they did more path training in third grade, they continued to improve in both reading speed, comprehension, spelling, and word identification. Now, you may wonder, what is the biological basis of this vision training? It has been shown that dyslexics have sluggish magnocellular neurons in the dorsal stream, yet normal parvocellular neurons in the ventral stream. We hypothesize that sluggish magnocells cause timing deficits between magnocells, which encode movement, where the word is located, and parvocells, which encode pattern information, analyzing the individual letters. Reading requires the synchronization of many functional systems. An imbalance between magno and parvocell functioning causes a specific deficit in reading. This deficit in reading from sluggish magnocells prevents the dorsal or motion pathways, where things are located, from working with the ventral or pattern pathways, what things are. Cognitive processing problems ensue because the where and what pathways are activated at the same time, and the what pathway does not know where to look. What happens when the pattern and motion pathways are not working together? Here are the word distortions that result from these sluggish magnocells. Dyslexics always go to the middle of the word, often not seeing the first letter, so they wouldn't see the W. They might flip the R and the O, flip the B and the D, and so that leads to confusion and misrecognition. So they see a totally different word than was in there. The dorsal stream sets up the frame of reference where the word is located, and the ventral stream analyzes the details sequentially. The dyslexic appears to tunnel down to get meaning, analyzing the individual letters, and does not know where to go next with the eyes since the magnocells are so sluggish. This is our working hypothesis. We propose that low-level deficits in the dorsal stream disrupt processing at higher levels. So if we go to the bottom here, we have, it is known that dyslexics have sluggish magnocells which disrupt processing in V1. These low-level deficits cause deficits in MT, medial temporal cortex, medial superior temporal cortex, which causes disrupted motion processing, deficits in lateral infraparietal cortex, frontal eye fields, which causes disrupted saccade planning, and deficits in dorsolateral prefrontal cortex, which causes disrupted central executive network. Moreover, remediation of these low-level deficits enable high-level cognitive functioning to improve dramatically, as now shown in collaboration with Dr. Ming-Shang Wang, the director of the MEG Radiology Imaging Lab at UCSD, who will be talking next about his really novel, incredible methods. So we have found that timing is everything. The where pathway must be faster than the what pathway to pay attention, read, multitask, and remember, to learn effortlessly, to have central vision, the letters in the word, and peripheral vision, signaling the beginning and end of the word, to work together. MEG source imaging in Dr. Ming-Shang Wang's lab was used to assess functional improvements in dyslexics following the path training. This was conducted at UCSD by Professor Ming-Shang Wang and his team of scientists. The subject was 29 years old, very bright, yet reading at a 6th grade level. We administered standardized reading, attention, and working memory tests before and after the path training. This subject did left-right movement training for 15 minutes, 2 times a week for 8 weeks. And wow, look at these improvements. We found many improvements. Using the MEG brain source imaging. Dr. Ming-Shang Wang at UCSD analyzed MEG source imaging evoked by a low contrast, 1 cycle per degree sine wave grating moving relative to a stationary 1 cycle per degree background. We found substantial increases. I'll just go here and show you. Substantial increases in V1, V3, MT, MST, that's ACC. So in both the dorsal network and the attentions networks, ACC, dorsolateral prefrontal cortex, ventrolateral prefrontal cortex, and the precuneus posterior cingulate cortex. Now, standardized tests that were administered before and after the visual movement discrimination training were consistent with improvements found in brain imaging. We found major behavioral improvements following visual movement training and working memory from the 6th to the 99th percentile, delayed recall, the 1st to the 25th percentile, reading speed, 154 words a minute to 437 words a minute, visual focus, 1st to the 54th percentile. This totally transformed his life. So in conclusion, this study provides strong evidence of functional changes following PATH training, consistent with behavioral changes in the dorsal visual processing network, attention network. So these advanced MEG source imaging for assessing functional changes with a 1 millisecond temporal and 1 millimeter spatial resolution in multiple brain areas is beneficial for assessing cognitive rehabilitation. And in summary, Path to Reading Intervention is a clinically validated program based on neuroscience research, improves reading fluency, attention, remembering, and multitasking by improving visual timing. Path trains the brain pathways to operate together, and it's simple and fast to administer. Thank you.